So in this video we're gonna learn about the kidney and the structure of nephrons as well. So let's start with the structure of kidney first of all. So externally we can see that kidney, they are the small bean shaped and reddish brown organs. Okay, so the kidneys are small bean shaped and reddish brown organs which are located in the abdominal cavity. They are in pairs. There is one pair kidney in human beings which is bean shaped, and the outer part of kidney is convex and the inner part of kidney is concave. If you draw the structure of kidney here, it will be somehow like this. So this inner part, so this outer part is convex, outer part is convex, and the inner part is concave, and this inner part, concave part, is called the hilum. Okay, this part is called hilum. This concave part is called hilum. Now through the hilum, the renal artery, it enters inside this, and the ureter along with the renal vein, it exits through the kidney. So renal artery enters and renal vein and ureter exits. The size of the kidney is approximately 9 cm into 6 cm into about 3 to 4 cm, and its weight is approximately 150 gram. Now it is located in the upper abdominal cavity on either side of the vertebral column. If we go for precisement, it will be T12 to L3, but this is thoracic, 12th thoracic vertebra and 3rd lateral vertebra, but this is not required. Just it lies on the upper abdominal cavity on the either side of vertebral column, and the position is slightly asymmetrical. It means that the left kidney is slightly superior than the right kidney. If the left kidney is here, then the right kidney is here because of the large size of liver on the right side. Now we'll go for internal structure of kidney, and that's basically what it's asked. So when we go for internal structure, we can see a figure which is this. So internally, kidney is divided into 4 parts. They are the cortex, the medulla, and the covering capsule, and this renal pelvis. So see, when we have to write about it, we'll write that the kidney is divided into 4 parts. The first of all is capsule. Now the capsule is the outer part of the kidney, which is the thick covering. It is the thick, tough covering of the kidney. Now just one line, and then we'll go for cortex. So this dotted part is called cortex. This outer part is called cortex. So the outer granular part of the kidney, which is about 1 cm thick, and it is dotted in appearance, that is called cortex. Now it consists of Malpighian bodies. While teaching about the structure of nephron, I'll teach you what are Malpighian bodies, what are PCT, what are DCTs. So it consists of Malpighian body, the proximal convoluted tubule PCT, and the distal convoluted tubule DCT. I'll teach you about these in the structure of nephrons. Then you'll understand how it is located in cortex, or how the loop of Henle is located in medulla. So this is the second part, and now third part is our medulla. Now medulla is the inner striated and darker part of the kidney. It consists of Henle's loop. Okay, it consists of Henle's loop, and it consists of collecting duct. Now if we see the medulla, this part is called medulla. If we see the medulla, it is divided into certain conical structures which are known as renal pyramid. So we can write it that the medulla is divided into a number of conical areas which are known as the medullary pyramid, and between the medullary pyramid, the substance of the cortex, it extends between the medullary pyramids to form the column of Bertini, or we can call it renal columns as well. And then at last we have the pelvis. Now pelvis is this part. So basically pelvis consists of a number of cup like structures which forms the major and the minor calyces, It collects the urine which is carried out by the ureter. Now each kidney is divided into a number of nephrons. Okay, and nephrons are made up of two parts. They are Malphigian bodies, or we can also call it renal bodies. Okay, and it is made up of nephric ducts. Now nephric ducts have a number of names. It can also be called secretory duct, and it can also be called as renal tubule. Okay, now further Malphigian bodies divided into Bowman's capsule and glomerulus, and nephric duct is divided into PCT, loop of Henle, and DCT. This is all about the loop of the kidney. Whenever we need to write about the kidney, we'll just write these four things. We'll explain them and make a figure of kidney. Now for ease, I'll show you how to make the figure. See, first of all, we'll make the bean shaped. The inner part, hilum, should be concave, and we'll take out ureter through it. Okay, should be something like this. Now this is the renal artery goes in, ureter and renal vein comes out. Okay, now this pelvis region is divided into a number of cup like structures to form the major and minor calyces. Okay, so this is our major calyx, and these are our minor calyces, and this is the renal pelvis. Now here are the medullary pyramids. Okay, so medullary pyramids will make, these are the medullary pyramids, and just draw a line on the edge of it and make a dotted appearance here. This will be our cortex, and between them make some blood vessels, and this will be our column of Bertini. Between them the column Bertini, and this whole will be our medulla. Okay, and this outer part is capsule, the covering. So this is all about the basic structure of kidney. Okay, histologically, and when we need to write, we can see that it's just one paragraph about the external structure which I said, the size, weight, color and location and all, and then the figure, and then only the capsule, cortex, the medulla and the pelvis. And just that each kidney is made up of numerous delicate nephrons. Each nephron consists of Malphigian body. It has cup shaped Bowman's capsule and a tuft of capillaries called glomerulus, and then it has secretory tubules and nephric ducts, which are proximal convoluted tubule, loop of Henle, and distal convoluted tubule. This is all about the kidney, and now you'll know what are these things when I'm going to teach you about the structure of nephron. So let's move on to the structure of nephron. So basically now we're going to read about the nephron or uriniferous tubule. Now if you see the chart, the nephron is divided into two parts: the renal body or Malphigian body, you can use any of the term, and another renal tubule, nephric duct or secretory tubule, you can use any of the term. For ease we'll use Malphigian body and nephric duct. Okay, it will be more easy. Now see, Malphigian body is further divided into Bowman's capsule and glomerulus, and this nephric duct is further divided into proximal convoluted tubule, loop of Henle, and distal convoluted tubule. So now we are going to read this structure in detail. Basically nephron, it is the structural and functional unit of kidney, and it is about 3 cm long. And for information, there are about 1 million nephrons in each kidney. So we are going to read about the structure. So first of all, we will write the Malphigian body. So it is divided into two parts. There, renal body, Bowman's capsule, and glomerulus. So see, first of all, we'll make the figure of the Bowman's capsule and glomerulus. So see, if we make the Bowman's capsule, it is somehow like this in structure. Okay, somewhat like a hook. Okay, for precisement I'll draw it here again. Okay, now see, it is a double walled structure. So this outer wall, it is made up of squamous epithelium, simple squamous epithelium. This is the outer wall, and similarly inner wall is also made up of simple squamous epithelium. Okay, we'll make the nuclei, and then just a simple thing, here is an arteriole which is known as efferent arteriole, and here is an arteriole known as efferent arteriole. This is the efferent arteriole, and this is the efferent arteriole, and we need to make the glomerulus here. This is our Bowman's capsule, and here is our glomerulus. Now the glomerulus are connected with each other. It means that glomerulus basically it is a capillary, and we know that capillary is somewhat like this. It is interconnected with each other, isn't it? So there is a technique of making the glomerulus. We'll make a triangle and then we'll make some like this. You can follow this. Okay, these are the glomerulus. If you see the figure clearly, it will be somehow like this. So this space is called capsular space. Efferent artery, the parietal layer means outside one wall, and visceral layer means inner wall. Now see, this is the figure. We'll make the figure first of all, and then we'll write what is the Bowman's capsule. So first of all, we have the Bowman's capsule. This is the figure of the Malpighian body. Okay, this is the figure of the Malpighian body. Now this part is called Bowman's capsule. This part is called Bowman's capsule. Now basically Bowman's capsule, if we write it point wise, it will be more precise and it will be easy as well. So it is a double walled structure. It is a double walled structure. See, double walled means this wall and this wall, double walled. Now the outer wall is known as the parietal layer. We always know parietal means outside and inner, visceral means inner. So parietal layer, which is made up of simple squamous epithelium, and then we have the inner wall. Inner wall is known as the visceral layer. Okay, and the visceral layer is also made up of simple squamous epithelium, but there are some podocytes in it. Now I'll explain what are podocytes. Now there are podocytes in it having cytoplasmic processes and pedicels. Okay, now I'm gonna explain what is this podocytes, cytoplasmic processes and pedicels. Basically podocytes are, so we'll see in the book. So if this is the capillary, okay, so see there are podocytes like this. This is basically the basement membrane. So these are the podocytes, okay, around the capillary, and these podocytes are extending to form this cytoplasmic processes. Now this whole is called cytoplasmic process. Okay, this one is called cytoplasmic process, and this single piece is called the pedicels. Okay, this part is called podocyte, this part is called cytoplasmic process, and this single one is called pedicels. Now basically we are not going to explain it in this nephric duct. So see, Bowman's capsule, double walled, outer layer is parietal layer, inner wall is visceral layer, and it has between the outer layer and the inner layer we have the capsular space. This space is called the capsular space. This is all about the Bowman's capsule, and the function we'll be learning in the formation of urine. Then another part we have the glomerulus. Okay, so glomerulus, they are the network of blood capillaries. They're the network of blood capillaries inside the Bowman's capsule. This network of blood capillary inside the Bowman's capsule, it is called the glomerulus. Basically glomerulus is formed by the efferent arteriole. So this is the efferent arteriole. It enters and forms the glomerulus, and all the glomerulus unite to form the efferent arteriole. So it unites to form the efferent arteriole. In this way we'll write certain points, and that will be enough for marks. So see, it forms efferent arteriole, and then there are large number of pores in them, called fenestrations. Now the pores are known as fenestrations. Okay, now these pores in the figures we can see the pores are in between them, and these are called fenestrations. Through these pores the blood goes inside the Bowman's capsule. We'll be learning it in the urine formation. Now it is embraced by the podocytes. As I said, it is embraced by podocytes. I'll show you how it is embraced by podocytes. And one thing, that this glomerulus, the network of blood capillary, it is more permeable than any other capillary. It is more permeable than any other blood capillaries. It means that it allows the blood to pass at a higher rate than any other artery. And one last thing, that the diameter of the efferent arteriole, this efferent arteriole, it is more than the diameter of the efferent arteriole. Now if we need to make the figure of glomerulus, we'll take a small portion of the glomerulus, and this will be the blood capillary. Okay, basically glomerulus is a blood capillary. We are taking a small portion. This is the small portion attached to this visceral layer. Okay, to this visceral layer, and we know that the visceral layer has podocytes having cytoplasmic processes and pedicells. Cytoplasmic processes are basically the extensions of the podocytes I showed you, and pedicells are the units of the cytoplasmic processes. Now see, podocytes are there. When it is attached to the visceral layer, the podocytes of the visceral layer, it attaches to this, and it forms somehow like this. Okay, from both the sides it attaches like this and forms this structure. Now this is called podocyte. This whole structure is called cytoplasmic process, and this unit is called pedicels. Now this glomerulus has number of pores in it. Okay, number of pores in it, they are called fenestrations. So this is the basic figure of glomerulus, and from in the notebook you can see the figure should be somehow this way. This is a diagrammatic structure, but if we need to make the structure of glomerulus for precisement, this is the precise picture of the glomerulus actually. Okay, so if you need to make the glomerulus, you can take the help of this figure. If you want, you can screenshot this figure if you do not find this in your notebook or textbook. Okay, so moving further, this is all about the Malpighian body. Now we'll go for the nephric duct, and we know that nephric duct, it is made up of PCT, loop of Henle, and the DCT. So basically nephric duct, it is a highly coiled structure. It is a highly coiled structure which is about 30 cm long, and it is surrounded by a network of blood capillaries called the peritubular capillaries. Now what this means is that okay, I'll make a figure, and then you will understand what are peritubular capillaries. So see, first of all, the glomerulus and the Bowman's capsule, we'll make them. Okay, so this is our efferent arteriole, and this is our efferent arteriole, and here is the glomerulus, and this is our Bowman's capsule. Now see, this part, it goes downwards, and it folds somehow this way. It folds, and then it forms a very thin tubule, and this tubule again loops like this to form a thick tubule. Okay, and then again it goes somehow like this, makes a fold. Okay, it makes a fold, and it connects to the collecting duct. Now this is the basic figure of the nephron. This is the entire figure of the nephron. Now see, this is our Bowman's capsule. Okay, this is our glomerulus, this is the efferent arteriole, this is the efferent arteriole. Now this part, this folded part, is called the proximal convoluted tubule, PCT. Okay, this part is called loop of Henle. This entire part is called loop of Henle. This folded part is called DCT, and this part is called the collecting duct. Now what this part, matlab itna upar and this much niche, so we'll make a line. Okay, now see, this part, it lies in the medulla of the kidney, and this much part lies in the cortex of the kidney. Now we understand that when we say that Malpighian bodies lie in the cortex of the kidney, we mean that this Malpighian body, it is lying in the cortex region. And as we said, as I said you in the structure of the kidney, that in cortex region, see, it contains of Malpighian bodies, it consists of PCT, and it consists of DCT. So see, it consists of PCT, it consists of DCT, and as I said that the medulla region, it consists of the loop of Henle and the collecting tubules. So see, it consists of loop of Henle, and it consists of the collecting tubules or collecting duct, whatever we can call it. It lies in the medulla region. If we see it on the basis of the structure of kidney, so we can see it making this way somehow. Okay, so this way, so this part, cortex region, this much lies in the cortex region, and in this medulla region lies the loop of Henle and the collecting duct. Okay, this is the figure of the nephron. Now we're going for the nephric duct, isn't it? So see, I said you that it is highly coiled structure, 30 cm long, and it consists of peritubular capillaries. Now what this means, that this efferent arteriole along with this PCT loop of Henle and DCT, this arteriole also goes downwards. Now in the similar manner as this is going, okay, in the similar manner, we will not make this in the figure. Still we have to understand that it is going in the similar manner. Now this efferent arteriole goes in the similar manner, and that is called the peritubular capillaries. It means that the entire nephric duct, this PCT loop of Henle and the DCT, it is surrounded by the arteriole which is known as peritubular capillaries, which is formed by the efferent arteriole. Okay, now see, we know it has three parts. They are the proximal convoluted tubule, first of all, PCT, then we have loop of Henle, and lastly we have DCT. So see, first of all we'll write about the PCT. So PCT, it is highly coiled structure, coiled or convoluted, you can use any of the terms. It lies in the renal cortex. Just write the structure, write the location in the renal cortex, and write what it's made up of. So it is made up of brush bordered ciliated epithelium. Okay, I'll tell you what this is. Ciliated, brush border, not ciliated. It is made up of cuboidal epithelium. Okay, epithelium. So it is made up of brush bordered cuboidal epithelium. It means that somehow like this. This is brush bordered cuboidal epithelium, and it consists of a number of mitochondria. Now mitochondria provides energy for active transport. Now what is the use of mitochondria in this, we'll learn in the mechanism of urine formation. So easily see, PCT, it is highly coiled in renal cortex. It is made of a brush bordered cuboidal epithelium. The loop of Henle is a U-shaped structure. It lies in the renal medulla. And now see, it has two limbs. If you see the figure, it has two limbs. This limb is called the descending limb, and this limb is called the ascending limb. It goes downwards and it goes upwards. So see, it has two limbs. They are the descending limb and the ascending limb. Now the descending limb, it is made up of simple squamous epithelium, and it is made up of simple cuboidal epithelium. I'll teach you how it is how to remember this brush bordered cuboidal epithelium, simple squamous and cuboidal epithelium. Now see, DCT, it is also a highly coiled structure. It lies in the renal cortex. The structure, the location, and then the formation, it is made by simple cuboidal epithelium. Okay, made by simple cuboidal epithelium. Now when you make the figure, for remembering, you can make somehow like this. See, this PCT, you can make that PCT is made up of somehow like this. You can make, this is the brush bordered cuboidal epithelium. Okay, the PCT is made somehow like this. And then if we go for the descending limb of the loop of Henle, it is made up of simple squamous epithelium like this. And if we go for the loop of Henle, it is made up of cuboidal epithelium, and DCT is also made up of the cuboidal epithelium. We can remember in this way, okay, by making the figure. Now the last thing, we have to write about the Vasa Recta, and we have to write about the duct of Bellini. Now I'm telling you what these both things are. Till now we did the Malpighian tubule or the Malpighian body. We did about Malpighian body, Bowman's capsule, glomerulus, and we went for the nephric duct, PCT, loop of Henle, DCT. Okay, and now the last thing, the Vasa Recta. I said you, after doing this, we'll just write a topic, the Vasa Recta. And what we mean by Vasa Recta, they are the peritubular capillaries. They are the peritubular capillaries around the loop of Henle. I said you that this efferent arteriole makes the peritubular capillaries around the entire nephric duct, but around this loop of Henle, the peritubular capillaries, the same peritubular capillaries, it is known as the Vasa Recta. Only this much we have to write. And about the collecting duct, we'll write that the DCT, it meets inside the collecting duct. Okay, see, this is one nephron. Okay, basically if we see the collecting duct, it is somehow like this. If we make the collecting duct, here one nephron meets, here another nephron meets, the third nephron meets here. Okay, so DCT meets here, another nephrons DCT meets here, another nephrons DCT meets here. So all this DCT meets in one collecting duct, and that large collecting duct is called the duct of Bellini. So it is the largest collecting duct. Okay, this is called the duct of Bellini, the largest collecting duct. So if we need to write about the structure of nephron, this much will be enough. Just describe about the Malpighian body, describe about the nephric duct, and describe about Vasa Recta and duct of Bellini. For duct of Bellini, again I am saying, see, the basic figure is this: proximal convoluted tubule, loop of Henle, DCT, and see it meets here, another nephrons meets here, and this large collecting duct, this is known as the duct of Bellini, and the Vasa Recta are the blood, the peritubular capillaries around the loop of Henle. So this is all about the structure of nephron. So hope you understood, and in the next video we will be discussing about the mechanism of urine formation. For now, thank you.